And now with our third video on the nervous tissue, we're going to discuss synapses, the connection between two neurons. We'll also be looking a little bit at how one neuron may affect another neuron, how the signal is both transmitted and what it does to the second neuron in a series. We're going to look at larger groups of neurons acting together, and then we'll also finish up with some repair processes associated with the neurons. All right, so there are two basic types of synapses. So remember, a synapse is a connection between two neurons. Right? A synapse could also be a connection between a neuron and a muscle cell. If the muscle cell is transmitting an electrical signal, then it could be a connection between two muscle cells. So in general, with all those different types of connections, there are two main forms of the synapses. The first is called an electrical synapse. An electrical synapse is where you have two cells connected together through gap junctions. So a great example of a chemical synapse would be between cardiac muscle cells. Cardiac muscle. So in a cardiac muscle cell, if we have one cardiac muscle cell here, the end of one cardiac muscle cell, and we have another cardiac muscle cell here, we could have these large openings or gap junctions in between the two cardiac muscle cells, so that if in one cell we started getting a high concentration of, let's say, sodium ions, those sodium ions will transport through those gaps into the second cell, and you'll start getting a high concentration of sodium in the second cell. All right. The movement of the sodium, those positive charges, are going to help depolarize the membrane. So that's how one cardiac muscle cell will send its electrical signal to the next cardiac muscle cell. That transmission will be two ways. It's always going to be going down a concentration gradient. Sometimes we do see electrical synapses in the brain where it coordinates large groups of neurons. The other type of connection is a chemical synapse, and this is the one we typically think of when you think of a synapse. In a chemical synapse, a presynaptic neuron, the neuron in front of the synapse, will release a chemical, a neurotransmitter, that will diffuse across the synapse and affect the postsynaptic neuron. Now there are three different ways you could get the connection between these neurons. So if I were to draw one here, so here's the end of an axon coming from the, so that's just the axon from the presynaptic. All right. It might contact the dendrite of a postsynaptic neuron. So if I were to draw these in, so it's attaching to the dendrite of this postsynaptic neuron, post after the synapse. All right, so that would be called an axodendritic connection. But sometimes the axon might directly contact the neural cell body. All right, that would be called an axosomatic connection. Soma is another term for cell body. And then finally, you will occasionally get a connection between an axon from the presynaptic axon will directly contact the postsynaptic axon. This is called an axo-axonic connection. These will help, could inhibit the conduction of the action potential, so it could stop the action potential from going all the way down the axon. All right, so let's look in a little more detail at chemical synapses. Now, this shouldn't be any surprise because we've seen this already in the muscle cell. So we went over a type of synapse in the muscle cell. We called it a neuromuscular junction. That's simply a synapse between a somatic motor neuron and a skeletal muscle fiber. So we have our action potential. It goes all the way down to the end of the axon to a synaptic end bulb. When the action potential, that change in voltage, goes all the way to the end, that's going to open up some voltage-gated calcium channels. So here we can see the calcium channel right here, step 2. Calcium is required for exocytosis. Calcium will activate some docking proteins on the inside of the cell membrane. So the next step we see is exocytosis. Down here at 3, we see some exocytosis. So all of these synaptic vesicles right here and here and here, are loaded full of neurotransmitter. They will dock, attach to the cell membrane, and they'll release the neurotransmitter out of the cell. The neurotransmitter will diffuse across the synaptic cleft. The synaptic cleft is the opening between the two cells, and it will bind to ligand-gated receptors on the postsynaptic cell. So here's in green are all, here in green is our listing right there of all of our receptors. So those receptors are also ion channels. They open up and will allow ions to pass into the postsynaptic neuron. The more neurotransmitter that's released, the more these receptors are activated, the more these ligand-gated receptors are activated, and the greater the difference in the postsynaptic cell. It's producing a graded potential. But we sometimes call this type of a graded potential a postsynaptic potential, because it's the graded potential that occurs in the postsynaptic cell. Right? Now this is only going to be a one-way transfer of information. So neurotransmitter will only go from the presynaptic neuron to the postsynaptic neuron. Right? If this form of a graded potential, this postsynaptic potential, is enough that by the time it hits the trigger zone, it hits negative 55 millivolts, we get an action potential. We get a nervous impulse. All right, so just like when we saw the graded potentials, I said that the graded potentials could be depolarizing or they could be hyperpolarizing. Well, it's the same thing here. So we could have excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So first let's talk about a depolarizing postsynaptic potential. We call them an EPSP, and that stands for E for excitatory postsynaptic potential. Right? Usually it's going to result from the opening of sodium channels. Right? So sodium will rush into the cell, and if we look down here in the bottom graph, this is an example of an excitatory postsynaptic potential. You'll notice it's also depolarizing. We start off at the resting membrane potential, we depolarize, and then we go back to a resting membrane potential. Okay, so when we depolarize, you'll notice this, it only takes it up to about 60 millivolts, but we're making it more likely to reach the threshold. So if you sum together many excitatory postsynaptic potentials, you'd be more likely to hit negative 55 millivolts because of this first one. But you can also have an inhibitory postsynaptic potential, also known as an IPSP, inhibitory postsynaptic potential. So this is going to be hyperpolarizing. So if you look up at the top graph, you can see that we start off at a resting membrane potential, then we hyperpolarize the membrane, and then it comes back to the resting membrane potential. Right? This is going to result from the opening of two different types of gates, either a chloride channel, either a chloride channel or a potassium channel. Okay? So chloride is negatively charged. Chloride is found in higher concentrations outside of the cell. Remember, sodium and chloride go together. So if we opened up these channels, that's going to allow negative charges to go into the cell. That's going to hyperpolarize the membrane. Or if we open up the potassium channels, then potassium will leave the cell because we always have a higher concentration of potassium on the inside of the cell. This will also hyperpolarize the membrane, so it causes it to become more negative or hyperpolarized. That means it's going to be less likely to reach threshold. So if we dip down from negative 70 to about negative 75, right, we are even further away from the threshold. So normally we would go from negative 70 to negative 55. That'd be a plus 15 millivolt change. If we hyperpolarize the membrane down to negative 75, we've got to have a plus 20 millivolt change. So we made it less likely to hit that threshold. So we call it an inhibitory postsynaptic potential. All right, so we have our neurotransmitter released from our presynaptic neuron. It will diffuse across the synapse and affect the postsynaptic neuron. We've got to be able to do something with that neurotransmitter. If you just leave it in the synapse continuously, you would just continuously stimulate the postsynaptic neuron, and you want to have good on and off type of control within the nervous system. So there are 3 different ways that a neurotransmitter can be removed, and we've already looked at a couple of these ways when we were looking at the neuromuscular junction in our muscle cells. So in the case of the neuromuscular junction, the neuron released acetylcholine. It went across the synapse, but it also could diffuse out of the synapse down a concentration gradient. So diffusion is one way that a neurotransmitter could be removed. We also saw in the neuromuscular junction the presence of an enzyme called acetylcholinesterase that broke down the neurotransmitter. So enzymatic degradation is another way that the neurotransmitter could be removed. Or the neurotransmitter may be actively taken back up by neurons or glial cells, and usually the glial cells involved will be astrocytes. So specific neurotransmitter transporters. So here's an example of how we can mess around with this. There is a neurotransmitter known as serotonin. Serotonin is often associated with feelings of well-being. All right? It regulates general moods within the brain. If you have the right amount of serotonin being produced, you feel as if you have a feeling of well-being. But if you are not producing enough serotonin, then you feel mentally depressed. In fact, that's a form of clinical mental depression, is the inability, not being able to produce enough serotonin. So serotonin is an example of a neurotransmitter that is actively taken back up by neurons or glial cells. So it has active reuptake. If you don't produce enough serotonin, it might be nice to block that reuptake, and that's exactly what Prozac does. Prozac is a serotonin reuptake inhibitor. In fact, sometimes it's called a selective serotonin reuptake inhibitor. Or you may have heard the term an SSRI. If you know someone who's struggling with clinical mental depression, they may be taking an SSRI. Prozac is just one example of them. All right, so we have our presynaptic neuron releasing the neurotransmitter, goes across the synapse to our postsynaptic neuron, and we have our postsynaptic potential. So there's 3 main responses you may get in that postsynaptic neuron. You could get a small excitatory postsynaptic potential, EPSP, excitatory postsynaptic potential. It is depolarizing, and it may take that neuron from negative 70 all the way up to negative 56 millivolts at the trigger zone. But if it does not hit negative 55 millivolts, you do not get an action potential. So it'll die out as an excitatory postsynaptic potential. But if it does reach the threshold, negative 55 millivolts, you get a nervous impulse. You get an action potential down the axon. If the membrane receives an inhibitory postsynaptic potential, IPSP, inhibitory postsynaptic potential, that means the membrane has been hyperpolarized, drops below negative 70 millivolts. Maybe it goes to negative 75 millivolts at the trigger zone. But in that case, you not only will not get an action potential, but you make it even less likely that you could get an action potential. So we've already seen summation. I believe it was in the first lecture. Summation is when you have several different graded potentials, several different postsynaptic potentials coming in either at the same time or maybe in sequence. So if several presynaptic end bulbs release their neurotransmitter about the same time, the combined effects could sum up and be enough for a nervous impulse. That could be several presynaptic end bulbs, several neurons coming in. That would be called spatial summation. Or you could have a single presynaptic neuron constantly releasing a little bit of the neurotransmitter. That would be temporal summation. So when we see this, here's a good example of spatial summation, the summation of the effects of neurotransmitters from several end bulbs onto one neuron. So you can see up at the top we have our two blue presynaptic neurons, both of them connecting with our third postsynaptic neuron. So if neuron 1 on its own just released one round of neurotransmitter, you can see by looking at the graph, that wouldn't be enough to take this to the threshold. If neuron 2 on its own released one round, then that wouldn't be enough to reach the threshold. But if you started adding together the effects of 1 plus 2, and maybe even more, it could sum up to being enough to reach the threshold. And then you would get an action potential going down the axon. Or, remember, you could also get temporal summation. So if you have neurotransmitters released from two or more firings of the same end bulb, so the same presynaptic neuron keeps releasing neurotransmitter once, twice, 3 times. If it's too slow, as you can see from looking down at the graph, the first 2 arrows are showing you where the release of the neurotransmitter and the effect dies out. It's too slow. But when they start coming really quickly, you could very quickly sum up all of the postsynaptic changes and finally hit the threshold. All right, so now we're going to look at a few different types of neurotransmitters. Some neurotransmitters will be excitatory. Some neurotransmitters may be inhibitory. You have both excitatory and inhibitory neurotransmitters present in both the central nervous system and the peripheral nervous system. And the same neurotransmitter can have different effects. In some locations it may be excitatory, but in other locations it'll be inhibitory. It just depends on what kind of ion channel it opens up. If you have the same neurotransmitter that opens up ion channels and they are sodium channels, then the effect is going to be excitatory. If the channels that open up are potassium channels, the effect will be inhibitory. And you can also modify those neurotransmitter effects. So when you think about all the neurotransmitters and the effects and what happens, you can change the effect along any one of the sequence of events. So first you have to make the neurotransmitter, then you have to release the neurotransmitter. You could remove the neurotransmitter from the synapse, or you could also block the receptor site. Now, we've actually talked about at least the last 3 of these. So release being blocked, we looked at the effect of Botox on a somatic motor neuron. Removal could be blocked. We looked at the selective serotonin reuptake inhibitor. The receptor site could be blocked. We looked at the effect of curare, which will block the receptor sites. Okay, so anything that will enhance a neurotransmitter's effect we call an agonist. Anything that blocks it is called an antagonist. We've used those terms before. Let's look at a couple of different types of neurotransmitters, including the most common neurotransmitter, acetylcholine. Acetylcholine is released by many peripheral nervous system neurons, including somatic motor neurons. It's also found in the central nervous system. It is often excitatory at the neuromuscular junction. That's the one we've looked at in detail. But in many other areas, it's inhibitory, and it gets broken apart by acetylcholinesterase. A couple of other small molecule neurotransmitters are modified amino acids. So 2 examples of amino acids. We have glutamate, which is a very simple amino acid. It's excitatory in nearly every neuron in the brain. Glutamate will activate the brain. There's another form of an amino acid called gamma albuteronic acid, or GABA. It's inhibitory for most of the brain synapses. And Valium is a good example. It is a GABA agonist, and that means it helps out the effect of GABA, and it makes it very inhibitory, and Valium usually makes you feel very sleepy or dopey. Then if you look on the blue side of this graph, you can see that there's all sorts of other different small molecule neurotransmitters that are slightly modified amino acids, including norepinephrine and epinephrine, dopamine, and serotonin. Now we're going to, in AMP2, look quite a bit at the effects of norepinephrine and epinephrine in the autonomic nervous system. Now here's the molecule GABA. Right? GABA is sometimes taken as a drug supplement. Right? But unfortunately, I've seen it in a lot of health food stores as a drug supplement, and the idea is that it helps you sleep. But if you take the supplement that only has normal GABA, and this is the type that's found in the brain, you've got a problem. It won't cross the blood-brain barrier. So if you ingest it, it gets into your bloodstream. It may circulate through your brain, but it's not going to get into their brain tissue. But there is an alternate form that is sometimes packaged and sold as a sleep enhancement called phenyl-GABA, or some variant like that, and it has this large benzene ring on it that will allow it to cross the blood-brain barrier. So it's very effective at assisting sleep. All right, and so that's going to be the end for this third video lecture. We will begin our 4th video lecture with neuronal circuits.